Hello and welcome everybody to my channel Omni Bioentrance, and today we will be discussing CUET entrance exam question paper for the subject Botany conducted in the year 2024. If you are new to my channel, please do like, share and subscribe. If you want still previous year question papers for various subjects of CUET, you can go to my channel Omni Bioentrance. In the channel playlist C for CUET and you will find them there. Now coming to the next question, the number of ATP required for the conversion of 1N2 to 2NH4 during biological nitrogen fixation. So here they have asked how much ATP is required for nitrogen fixation. It is 16 ATP molecules. So answer is 4. Then lignin is a polysaccharide. No, lignin, it is a polymer of aromatic alcohols. And three main monolignals that are involved are the para-coumaryl alcohol, coniferyl alcohol and synapyl alcohol. So it is not a polysaccharide, so A is not correct. Is the second most abundant organic compound? Yes, that's correct. So the first abundant is cellulose. This is the second abundant. Is a polymer of aromatic alcohols. That is also correct. Synthetic pathway is peroxidase dependent. That is also correct. So B is correct, C is correct and D is correct. That gives your answer as option 4. B, C, D only. Then jasmonic acid. Okay. Jasmonic acid is synthesized from linolenic acid. Yes, that's correct. Is a growth promoter. It is not a growth promoter. It is involved in inhibiting plant growth and promote senescence. Delays leaf senescence. No, it promote leaf senescence. Present in mosses and ferns. Not yet studied. Because jasmonic acid, they are telling that it is present mainly in the angiosperms. So the answer here is synthesized from linolenic acid. The next one, the enzyme which converts citrate to isocitrate in the TCA cycle is aconitase. Answer is option 1. The amount of DNA present in the genome of a species is called its C-value. Which of the following is the most common form of DNA? It is your bDNA. All these are direct questions, that's why I am not explaining much. Then which of the following is true for about a poly-A tail? So the poly-A tail, you all know it is added to the 3' end of the mRNA molecule during the processing of mRNA in the nucleus. That is poly-A tail. And usually around 150 to 250 nucleotides are added. Nucleotides, that is adenosine nucleosides, are added to the mRNA molecule. So synthesized post transcriptionally, that is correct. Usually shorter than 300 nucleotides. Yes, I told you it is around 150 to 250 nucleotides, so that's also correct. Its function is to protect mRNA from cytoplasmic RNases. That's also correct. Its function is to stimulate translation. That is also correct. So A, B, C, D are correct. Answer is option 3. Then next one. VNTR sequence, variable number of tandem repeat. So that is involved in DNA fingerprinting. So A3. So it is seen in 2 as well as in 4 options. Whereas 1 and 3, it is A1. So we will see 2 and 3 which is correct. Then leader sequence, yes, it is functioning in attenuation. If you remember your tryptophan operon, you can find the leader sequence there. And the leader sequence, it is involved in attenuation reaction. So B4. So in 2 and 4, B is 4. Okay, we will see C. SD sequence is your Shine-Dalgarno sequence. And what is it they have given? Ribosome binding site. Yes, C2. Ribosome binding site. That is where the ribosome binds. So C2 is the answer. Then cis acting sequence, that is gene regulation on the same chromosome. So D1. So the answer here is A3, B4, C2 and D1. So that makes 2 as the right answer and not 4. So answer is option 2. Then next one. Euchromatin is transcriptionally active. Yes. And contains most of the protein coding DNA. That's correct. A is true. Heterochromatin is light staining. No. Heterochromatin is having more condensed DNA and it is dark staining. And less condensed portion of the chromatin. No, it is more condensed portion of the chromatin. So A statement is correct but R is wrong. So that makes answer 3 as the right answer. A is true but R is false. So 3 is your answer. Then solenocysteine is an amino acid which is present in only prokaryotes. No, it is present in eukaryotes also. So that is not correct. Coded by UGA. Yes, UGA, which is a stop codon, is used to code solenocysteine when specific recognition sequences are present in the mRNA. So 2 is correct. Derived from cysteine, no. Even though they are structurally similar, because in cysteine sulfur atom is present, but in solenocysteine it is selenium that is present instead of cysteine, but it is not derived from cysteine. It is a different pathway for solenocysteine. Is present in many eukaryotic proteins. Not only eukaryotic, it is present in prokaryotes also. So 4 is also not correct. So answer is option 2. Then given below are 2 statement, assertion, reason. Osmosis is a special type of diffusion in which movement of substance occurs from higher solute concentration to lower solute concentration. That is 100% wrong. That is not the correct definition of osmosis. Osmosis is the movement of water molecules through a semi-permeable membrane from a region of low solute concentration to a region of high solute concentration. That is osmosis. Okay. And this particular osmosis, it continues until the solute concentration on both sides of the membrane are equal. So A is not correct. Diffusion is a dominant mode of transport at the scale of a cell. That is 100% correct. So that makes your answer A is false but R is true. So that is option 4. Then again assertion, reason. The application of transgenic plants are in delayed ripening, production of bioplastic, plant as bioreactor for the production of antibodies, etc. Yes, this is the reason why you use transgenic plants. This is one of the reason. So these are the applications. So A is correct. Agrobacteria that carries a recombinant plasmid with both a selectable marker and a desired transgene are used to make transgenic plants. That is correct. And that is the reason why you are developing transgenic plants, because there is agrobacteria which is a natural genetic engineer to transform plants. So agrobacteria, it carries a recombinant plasmid because agrobacteria is having Ti plasmid. The Ti plasmid is made recombinant. How? By introducing a selectable marker gene so that you can select for transformants after transformation, and then you introduce your desired gene of interest also. So it is made recombinant and then you use it to transform plants. So R is the reason for correct reason for assertion A. So both A and R are true and R is the correct explanation of A. So 1 is the answer. Then the role of ethylene is senescence of leaves and fruit ripening. Yes, that is correct. 1 is correct. Then match the following. Allium sativa, plant source. It uses in insecticidal properties. So A3. A3 is there only in one option. So that must be your answer. A3, okay. B. Coptis japonica, it is antibacterial properties. So B4. Taxus baccata, it is again in anticarcinogenic. And digitalis lanata, it is cardiac medicine. So D2. So A3, B4, C1, D2. So this question shows that you have to be thorough with your economic botany. So B3, A3, B4, C1 and D2 is the answer. Then the shape of chloroplast in Spirogyra, it is spiral. That is why it got the name also. So A2. Then which of the following algae are used as biofertilizers? It is option C. If you see, that is the answer. Anabena, Azolla, Nostoc. I don't know why Azolla came. It is not a algae. Anyway, it might be a printing mistake. They may be planning to write as Anabena, Azolla. Okay. Now whatever, Anabena, Azolla and Nostoc is the answer. Name the fungus whose extract from the sclerotia can be used to produce a powerful hallucinogenic drug. It is your Claviceps purpurea that is producing this hallucinogenic drug, and it is called as ergot. So answer is option 2. Who discovered the heterothallism in fungi? It is A.F. Blackesley. So answer is option 1. Then match the following. Penicillin. Penicillin is obtained from Penicillium notatum. So A3 is there in 2, 3 and 4. Okay, fine. So we will see which one is correct out of that. A3. Then fermentation of sucrose and molasses to produce citric acid. So citric acid production, it is Aspergillus niger. So B1, that makes you to write off 4th because it is B4 they have written there. So that is not correct. So compare 2 and 3. Fermentation of carbohydrate to produce ethyl alcohol and carbon dioxide, that is your ethanol production, that is by Saccharomyces cerevisiae. So B4, that makes answer as option 3. Irish potato famine, the causative agent is Phytophthora infestans, that is D2. So A3, B1, C4, D2 is the answer. Then the alternate host for Puccinia graminis is, that is Berberis vulgaris, B4. Then next one. Which of the following is a prophylactic method of plant disease control? Eradication of alternate host. So that is one of the method because it aims to prevent the spread of the disease by removing a plant on which the pathogen will rely to complete its life cycle. That is called as alternate host, because alternate host is needed there for the pathogen to complete its life cycle. So once you eradicate the alternate host, then it is a preventive method. Then spraying plants with the pesticide and fungicide on a regular basis, that is not a correct method because it aims to control the disease symptoms only after when the disease have appeared. It is not a preventive measure. Okay, so B is not correct. Then crop rotation practices to control soil-borne diseases. Yes, because crop rotation, it reduces the number of pest and pathogen in the soil, such as when you plant resistant crops after a season of growing susceptible crops, the pathogens will be starved of the food. So that way is one of the reason why crop rotation can be used. Then growing antagonistic plant. Antagonism means when one organism inhibits or slows down the growth of a plant disease causing organism Okay, that is called as antagonism. So here what they have told is when you grow antagonistic plant around a susceptible plant, it creates a protective environment around the susceptible plant. Okay, so D also can be considered as a mechanism for a prophylactic method or preventive method of plant disease. Can be considered, but more apt will be A and C. So 2 is the right answer, that is A and C only. Then next one. Bryophytes are distinct from all other land plants in having an independent gametophyte and a dependent sporophyte. So 3 is the answer. Pseudo-elaters which help in dispersal of spore are present in anthoceros. So 4 is the answer. Then which of the following is not an identifying feature of cycas? It has anthrozoids that are motile, yes. Anatomical features of the leaflet indicating it as a mesophyte, no. That is the, they have answered because they have asked not an identifying feature of cycas. So 2 is the answer. Circinate venation, that is cycas characteristic. Xylem lacking vessel, cycas is a gymnosperm, so naturally no vessel. So 4th is also right. Then which of the following gymnosperm possess winged pollens? Dandelion. Dandelion is not a gymnosperm, it is an angiosperm. So first option itself is not correct. Cycas does not, is a gymnosperm, but it does not possess winged pollen. But Pinus is having winged pollen grains. So Pinus is the answer. Ephedra also is not having winged pollen grains. Then next one. A stele which does not contain a central pith is called. So solenostele, that is these two. Ectoploic solenostele and amphiploic solenostele. So ectoploic solenostele is having a pith, amphiploic solenostele is also having a pith. Amphiploic means it is having phloem on both sides of the xylem. So that is amphiploic. So that is having pith. So this is the case of solenostele. Dictyostele, again you can see a prominent pith there in dictyostele. So that's also not correct. Protostele. Protostele doesn't have a pith. It is having a cortex, endodermis, pericycle, leaf trace is there. Then xylem and phloem are also present, but pith is not present. Then siphonostele, this is the siphonostele, that is ectoploic siphonostele and amphiploic siphonostele. So they both have pith. So they have asked which is the one which is not having a central pith. So that is your protostele. Answer is option 3. Then next one. Which is the major secondary metabolite present in clove bud? It is eugenol. Answer is option 2. Thymol, it is a monoterpene phenol found in Thymus species. Then zingiberine, it is a sesquiterpene compound found in ginger. And piperine, it is an alkaloid present in black pepper.